Guarda che accendo la fiamma. Good morning. Ok. Good morning, students. Today we will start to go a little bit more into the details of a very significant question whose answer is not at all clear and definite anyhow. The question we would like to answer today is what is life? I know it is quite intriguing because we will discuss of several subjects but may be quite far from the experience of environmental engineers. In order to answer to these questions, and as you will see, we will come out with a book of Erwin Schrödinger, whose title is exactly What is Life? It was in 1944. In order to answer to this question, we shall go back a little bit to what we have seen already last week concerning energy flows at the level of the biosphere. As I already told you, the source of all energy transformation in the biosphere is the sun. So it is quite interesting to see how the energy coming from our star is subdivided while it arrives on the surface of the planet. If we make 100% the incident radiation on the surface of the planet, and I recall you when I'm thinking to this 100%, I'm thinking to 1,360 watts per square meter, that is the solar constant, that is an indication of how much the sun is working. And if we make 100% with 1,360 watts per square meters, then of this 100%, 35 is immediately reflected by dust, gas, clouds, and albedo. And then 90% is absorbed by atmospheric gas like ozone, steam, clouds, and so on. Only 36% of this energy arrives directly or indirectly on the surface of the planet, and it is then the basis of all the energetical transformations that appear at the level of the biosphere. Can you hear me? Excuse me, I'm just, somebody can give me a feedback if you are hearing me?
Yes, Professor, we can hear you well.
Thank you very much. Okay. We've also said that the energy transfers at the level of the biosphere as an efficiency, and we've discussed a little bit about the efficiency of these transfers. We simply have converted the 1,360 watts per square meters solar constant into kilocalories per year. And this 1,360 accounts to an amount of 1.2 10 to the 22 kilocalories per year. Kilocalories per year. So this is the flow, energy flow, yearly energy flow arriving on the surface of the planet. Of this energy, terrestrial vegetation and phytoplankton, what we shall call later primary production, use 2.5%, 2.5 10 to the 20 kilocalories per year in order to produce 5.2 10 to the 70 kilocalories per year of biomass. And if you make the ratio in order to calculate the efficiency, you end up with a figure of 0.2% efficiency of transformation. And this is the efficiency of transformation of solar light into primary production. It means phytoplankton and terrestrial vegetation. The efficiency is quite low, but as we go up with the trophic levels, so passing from the primary production to herbivores and carnivores that have higher trophic levels, you see that the efficiency becomes higher and higher, up to carnivores, or maybe us humans, but we are omnivores, can go up to 40% of efficiency. I've already introduced a series of concepts like production. Production in our context is photosynthesis, primary production, synthesis of organic molecules. The complementary process is consumption, that in biochemistry is called fermentation or respiration. These are oxidation processes. During consumption, ATP is produced, ATP molecules. It is the fuel for cellular processes. I recall you that ATP transport energy into between the cells. And by very simple measurements, calorimetric bomb measurements of energy content, we end up with these figures that are coefficients of energy content for different species of biomass. For example, we may subdivide biomass between plants, algae, invertebrates, or vertebrate biomasses. And the energy content are the figures that I am showing now. We can also subdivide the different types of biomass into different types of biochemical molecules, so carbohydrates, proteins, and lipids. You obtain similar figures. And this allows us to convert easily between weight and energy content. In order to understand how we end up to the situation we are experiencing now on the surface of the planet, may be useful to make us more recall or a brief history of the biosphere in order to understand and to appropriately collocating the correct historical framework, the story of evolution of the planet. So we may distinguish three ages in the evolution of life on our planet. The prebiotic ages, that goes from 4.5 to 3.5 billion years ago, so before life appeared on the planet. And now we have traces. We know that life should have appeared, or it appeared on the planet around 3.5 billion years ago. After this, we have a microcosm from 3.5 to 1.5 billion years ago. The microcosm era is the period in which bacteria, or small living organisms, and when I'm thinking of small living organisms, I have in mind bacteria, phytoplankton, things like that, dominate the planet. And please note that these are 2 billion years in which only bacteria, our friends, the bacteria, dominated the planet. During the microcosms, bacteria have the opportunity to change the condition, I mean physical-chemical conditions of the surface of a planet in order to foster or to favor the appearance of more sophisticated or more higher trophic level life forms. So after 1.5 billion years ago, we identified a macrocosm age from 1.2, 1.5 billion years ago up to today, and this is the era, a period in the story of the planet on which all superior life forms appeared. Please note that while the bacteria dominated for 2 billion years life of the planet, and we have quite a short 1 billion years time span in which the evolution produced higher forms of life. We shall realize that bacteria have changed the atmosphere during these 2 billion years, adjusting, for example, adjusting the levels of humidity, hydrogen, CO2, oxygen, and ozone, and so on. Please note that several of these elements, like hydrogen or CO2, or even oxygen, may be toxic or may be dangerous for life. In particular, this type of regulation is very, very precise, keeping, for example, the concentration of oxygen in the atmosphere 21%, and this is exactly the limit below which oxygen respiration wouldn't be possible. So 21% is the limit at which oxygen respiration, so oxidation, in terms of oxygen of organic matter. Above this concentration, so if we would have more than 21%, then the planet will be a place, a kind of nightmare, or if you want, a place where life would not be possible because, however, 21% we would have self-combustions. So you know that oxygen is a fundamental component in order to allow combustions. If we have percentages over 21% of oxygen to the atmosphere, then combustion will be very, very easy. So what we would say is a Navy-like place in which we will have fires everywhere, all over the time. So just to put it in the right perspective, the story of the planet, we have made a simple game of projecting, a linear projection, the various phases of the evolution of life on the planet onto a 24 hours clock. So we may subdivide first 12 hours, that is the night, and the second 12 hours, that is the day. So if we make a linear projection on of these times on this hypothetical clock, we see that 4.6 billion years ago it is the birth of the Earth, then bacteria appeared 3.5 billion years ago. So if we put at midnight 0 hours, 0 minutes, 0 seconds of the birth of the Earth, bacteria appeared at 5:44 in the early morning. And then photosynthesis was invented. Water photosynthesis was invented not earlier than 11 in the morning, 11 a.m. Then we had an important step in the evolution, the appearance of eukaryotes, bacteria, very sophisticated machinery, but by the employing of the symbiosis process, made a very important step forward along the evolution. And these eukaryotes appeared around noon, half past noon. Then we had to wait the whole day until 9 in the evening to see the appearance of invertebrates. So between half past noon and 9 p.m. in the evening, we may observe an incredible number of life forms appearing on the planet. In particular, these are life forms that developed in aquatic water ecosystems. So invertebrates, and when I'm thinking about invertebrates, I'm thinking about flatworms, are appearing 9 p.m. in the evening. This is another way of recalling you, as I already said some last week, that we study water ecosystems because water ecosystems are those places in which the most incredible or accelerated development of life forms appeared on the planet. So water ecosystems are really prototypical in order to understand how evolution took place on the planet. So we said invertebrates 9 p.m. The first vertebrates some seconds after 9 p.m. Then first plants of land, that is colonization of inland. This is around 10 p.m. Origin of reptiles, amphibians, fishes, and so on around 11. Insects appear at 10 p.m. Superior reptiles, and here I'm thinking about lizards and the ancestors of dinosaurs, appeared a quarter to 11. At 11 p.m. we have conifer, mammals, birds. So you see, very short time spans after the appearance of the first life forms, we have an explosion of a lot of different life forms appearing in a very, proportionally speaking, very short time span. So we have to wait until 11:48, so 10 to midnight, to see the appearance of mammals. and hominids appeared just 3 minutes before midnight in our projection, and Homo sapiens 1 minute, and the history as we know it today is just very few seconds before midnight of the, before midnight in the evening. So you see, I made also projections on these pie graphs of the story. You see that for a large part of the time since the birth, there was no life on the planet. When as life took place, we have an important, a significant part of time in which bacteria dominated the land, and then bacteria invented water photosynthesis, that is an important technological or if you want developmental step forward. And from this moment on, we have an incredible various uh, evolution of various life forms, starting from the eukaryotes. Aerobic respiration dominated an important part, and then the highest part of the trophic chain developed some minutes before midnight. So the impressive part of the story is that even if evolution had a long-term time to play in order to propose new technological or biotechnological developments, human life appeared very shortly. So we are here on a very short span. The incredible point, the incredible aspect of this is that we, in our very short persistence on the planet, we have significantly destroyed or changed in a dramatic way the condition on our planet. It is interesting, in order to see or to have an indication of how these changements were significant, to look at the composition of planetary atmospheres. So here we have a list of gases: CO2, gaseous nitrogen, oxygen, argon, methane, and so on, and some indicators like temperature or pressure. And we look at these values for the different planets. For example, Venus, Earth without life. Earth, this is of course a modeling projection, what it would be if we wouldn't have life development. Earth as it is today, so with life, and Mars. So just to start looking at CO2, that as you can imagine or may know, is an important indication of how the condition of the atmosphere are in the planet. You see that Venus, it is our closest planet in the direction of Sun. So Mercury and Venus is the second planet, and then we have the Earth. Venus has 96% CO2 in the atmosphere. Mars has 95% CO2 in the atmosphere. Earth, that is in between Venus and Mars, actually has just 0.03% CO2 in the atmosphere. If life wouldn't be there, then we would have 98% CO2. It is interesting also to look at oxygen. Only traces in Venus, very few oxygen in Mars, almost zero oxygen in Earth without life, and nowadays figure 21%. Nitrogen, the same, 79%, very few in Mars, very few in Venus, and there wouldn't be nitrogen or very few nitrogen in the Earth without life. So this just to explain or just to underline the fact, stress the fact that life has changed significantly the condition of on our planet. So significantly that we can see very different atmospheric composition between the various planets, and in particular, the atmospheric composition of the actual Earth is really specific or the only or very important, significant in order to see the development of life forms as we see them today. You can also notice, looking, for example, at our physical-chemical parameters like the temperature or the pressure. Temperature are terrible on Venus and are terrible on Mars. So every very hot Venus and very cold Mars. On the contrary, on the Earth, as I already told you last week, due to the Earth thermal machine, you have 14 Celsius degree. 14 Celsius degree as an average on a whole year and on the whole surface of the planet. With the pressure the same, we have one atmosphere, one bar of pressure on the Earth, while we have a very low pressure on Mars and terribly high pressure on Venus or on the Earth without life. So you see, just this table, these figures, just to show you how important was the changement of the condition of the planet due to the appearance of living forms. And I recall you when I'm talking about the appearance of living forms, I have in mind bacteria, our friends with bacteria, that have changed significantly the condition of the planet, and that they had, I recall you, 2 billion years in which they dominated the planet. So for these 2 billion years, there were only mainly bacteria on the planet. Okay, so let's go back a little bit to the questions where we started with lecture. What is life? What is life? Is an intriguing question. As I told you in the beginning, nobody yet knows exactly what life is. I would try to give you some definition, or I would try to propose you a way of defining life. In order to introduce this subject, it is important to notice or to stress a very important booklet, a small book that was published in 1944 by Erwin Schrödinger, which was one of the founders of quantum mechanics, by incidence, whose title was exactly What is Life, in which he tried to give some observation or to discuss or give some hints concerning the problem, concerning the appearance of these strange things. This is quite impressive, especially from a physicist's point of view, because a living object seems, as I already told you, to defy the second principle of thermodynamics. I recall you the second principle of thermodynamics, the so-called law of entropy, says that a system, an isolated system, if left to evolve alone, it will evolve towards states or will evolve towards states of maximum entropy. So the principle, the second principle of thermodynamics tells us that the entropy of an isolated system should increase unless something different takes place. But I'm saying isolated system means a system that is left alone to evolve. When I'm thinking to these systems, I'm thinking to thermodynamics, the thermodynamic paradigms for studying physical systems. In thermodynamics, we have two main principles. The first one is the principle of energy conservation, first law of thermodynamics, while the second is the principle of entropy increase. So the second principle of thermodynamics. Impressively, Schrödinger, while introducing this discussion, stated or placed on the table two instances. The first one is order from disorder. So he realized that in some controlled condition, some disordered structures may produce some kind of ordered system, and this seems to be contradiction. And this is exactly why physicists felt that this was a defy of the second principle of thermodynamics, because according to the second principle of thermodynamics, you may recall that order means low entropy, while disorder means high entropy. So the idea that high entropic system, so very disordered system, may manifest some kind of order in the structure is somehow intriguing, at least for physicists, less intriguing for biologists who observed on their everyday lives this fact. For example, as I already told you, made you an example. If you look outside your windows, you look, for example, at a tree or any other living structure like house, bees, and so on, you may recognize that there are some kind of ordered structures that are replicated and replicated somehow. So that living system seems to, at least locally, to reduce entropy, to build ordered structures. But that was more even impressive, the second instance that made Schrödinger. I recall you that this book is 1944. It is a very, very light reading, so I suggest you for this summer, next summer or this winter, if you don't know what to do in Christmas, to read this book. Very light reading, very pleasant. The second instances that was recognized by Schrödinger was what the one he called order from order. He said, or Schrödinger anticipated, that given the fact that all these structures that appear in living system had all a very well-defined building strategy, or well-defined building plan, Schrödinger suggested that somewhere it should have been written, or somewhere should have been recorded the way of building these structures. And he suggested that there should be some molecules somewhere that recorded somehow the way of building the structure. And I recall you that this is quite impressive because it is just 10 years after 1954, with Watson and Crick discovery of DNA, in which it was discovered that effectively such a structure existed, and was a structure formed by very particular biological molecules, so-called chromosomes. The chromosomes are, biologically speaking, proteins. And the fact of recording the structure for building structures into chromosomes was one of the more important tools for the evolution, for the Darwinian evolution. So we had the first scientific proof of the fact that Darwinian evolution of 1 century before, that was introduced 1 century before, somehow may be explained by what biologists today call molecular biology. Molecular biology is the study of DNA today. In order to explain how evolution may have been playing with molecular biology, so with DNA or with chromosome, in order to build new structures, there are several statistical considerations to be made. The point is that, let's say, evolution had a very temporal perspective, very long temporal perspective, what biologists or evolutionists call the deep time. So it is a matter of a lot of time, and we are talking about billions of years, I recall you, in which evolution had time to play a game of trials and errors and variation and trials, errors and changement, and this variation, what we would call to the mutation, are variation of what? Variation of the genetic code, chromosomes, and these variations are significantly induced by, for example, radiation. Hard radiation, what physicists call R radiation or X radiations or cosmic radiations. So that this is from a cosmological point of view, this is the cue of the Big Bang event, which left a kind of I would say a kind of archaeological print that is very cosmic radiation, and all these cosmic radiation interacts with living forms, in particular with molecular biology, so DNA, we're making mutations, and then the game of evolution make only survive those mutations that appear to be more fit or more adapted um, to the current condition of the environment. You may recall that we already introduced some of these concepts, like the concept of when we were discussing about ecosystems. You may remember that we said that ecosystems are systems that uh, try to maintain or make persistent lives according to condition of the surrounding environment. So living systems are systems that are able to adapt to the surrounding environment, to absorb transients, maintain constant conditions so that life has the time to foster and to develop. It is important also to recall that one of the aspects in the definition of ecosystems that we have introduced is that of the building of a construction of borders in order to uh, give a consistent definition of what is an ecosystem. We had to distinguish between inside and outside. So the important part here, but we may translate in if we want a scientific hypothesis, is that in order for life to appear, there should have been the possibility of building some kind of membranes or borders that distinguished an inside stable and constant from an outside that was random and unconstant. So that inside the conditions were preserved so that life could, or biological reactions, if you want, or biochemical reactions, may take place without so much perturbation. This is the theory of Varela's Mces that was developed in the late 80s. And the idea is that in the primeval soup that dominated for 1.5 billion years the surface of our planet, so we have a soup of more or less warm, not hot, but warm broth soup, in which we have all the molecules or all the elements necessary for building living biomolecules, or for building biomolecules, sorry. In this condition, due to the effect of external radiation, cosmic radiation, I tried to synthesize with this H nu, build some kind of distinction between border cells and something that was inside. So this is a first example or a first hypothesis to explain how living objects started to distinguish the environment, to make a distinction between what is inside, stable and constant, and what is outside, what is random and perturbed. So one of the aspects linked to the definition of what are ecosystems is that of building membranes or borders. And it is quite interesting if we look at the biology of the first living forms. On the left part of this slide, you have prokaryotes, bacteria, so the first forms of bacteria. You see prokaryotes just have a border, and inside pieces of, in the cytoplasm, pieces of nucleotides, so that is pieces of DNA, and a kind of interaction between the cytoplasm, so what is inside and what it is outside, through this structure that here I have indicated, like pilies, membranes, and so on. This kind of subdivision of space and time is also more evolved toward the eukaryotes types of bacteria that are more sophisticated bacteria that appeared later on. I don't know, let's go back just to give you, sorry, an idea. Until here, so until 2.2 billion years ago, we had only prokaryote bacteria. So prokaryote bacteria dominated from 3.5 to 2.2 billion years ago, and after 2.2 billion years ago, eukaryote bacteria appeared. Eukaryote bacteria are more sophisticated, where they present internal structures, organelles, and the evolutionary mechanism identified, for example, by Lynn Margulis was that of symbiosis, so different bacteria capable of doing of performing different biotechnologies stuck together by symbiosis and ended up in more sophisticated structures, in which we have, for example, a nucleus. Karyotes in Greek means nucleus. Eukaryotes means really nucleated cell, so a cell with a true kernel. And impressive aspect of this story is that, in particular, we find that the DNA, the chromosome, what was thought to be the biological intelligence, in these more evolved living objects like eukaryote cells, may be found principally in the nucleus or in the kernel of a cell. I recall you that in the prokaryote instead, we have pieces of DNA just mixed, welled up like in a blender into the cytoplasm. Like instead, in eukaryote structures or eukaryote cells, very well defined structures in which you will find what biologists call biological intelligence of information of the reconstruction of the system, in particular the nucleus. In the nucleus, we found the nucleus, and inside this nucleus, you find the DNA, in particular on the chromatin, that is the chromosome. It is very interesting also to note, and maybe we will have time to discuss this, but also there are different structures like the mitochondria. It is here, but are, if you want, the power plant of the eukaryote bacteria. So mitochondria are organs that produce ATP, essentially produce molecules with a high energetic content, and essentially they convert solar light into energy stored into chemical, biochemical bonds. It is quite interesting, but also in the mitochondria we have some portions of DNA, it is so-called mitochondrial DNA, but this DNA has a particular history. So it is a DNA that is transmitted only through the female from the mother to the offspring. So it is a DNA that is transferred only through female lines. So to make, let's conclude this lecture by making a recall of all the concepts, very weird concepts we have introduced today. We started by asking ourselves, what is life? We answered that life has to do with the evolution of different systems, and this evolution, some kind of information management where was apparent, and we may call this information management from one side intelligence, so understanding or linking different hierarchical levels through the management of information. We may call also this information management knowledge. So this knowledge is information that is stored. So if you look at the epistemological development of this branch of science, we start from the dogmatic thinking, creationism. You may recall some centuries ago, a very famous Italian physicist, Galileo, risked to be burned because he was just saying that the Earth was not the center of the universe. And with the same approach, creationists introduced the role of a divinity in order to explain evolution. But when we have, thanks God, in the end of the 19th century, Mr. Darwin, Charles Darwin appeared and introduced his evolutionary perspective, introduced the role of natural selection in order to explain how different life forms evolve on our planet. So this was evolutionism, natural selection, but then we had to wait 1 century more in order to understand that natural selection acted on a particular form of biochemistry, biochemical molecules, DNA, and this gave rise to the discovery or the introduction of a new branch of biology, the so-called molecular biology, introduction of genetics. Today, we are in a more modern perspective. We may call a neo-Darwinism perspective, but is the modern synthesis between evolutionism and molecular biology. So we simply erase or cancel creationism. We observe evolutionary processes. Evolutionary processes act by changing molecular biologies, genetics. These changes are induced by external perturbances. And so the modern synthesis of neo-Darwinism may be resumed by asking if it is really DNA, so the genetic code, biological intelligence. This is an intriguing question because for several decades, biologists were very dramatically concentrated on the understanding of the structure of DNA, with the dream for one chromosome, one protein. But thinking that the intelligence is linked to the simply to DNA would be resumed by saying that we are thinking by gonads. You may recall that DNA is the core molecules around which gonads that are earlier the first cells in the evolution of complex organisms are developed. So if we think that the biological intelligence is all including the DNA, we shall then be happy to understand how DNA is structured. The dream of a biologist, a molecular biologist, as I told you, is one gene synthesized or one protein. So we had the Genome Project in the late 70s, beginning of the 80s of last century. We recognize, we know that more or less in living system, we have 100,000 proteins. So we would be happy to identify at least 100,000 genes for explaining how life forms develop. Unfortunately, if we make some analysis, we discover that a fly is made up only of 25,000 genes, a worm 10,000, a bacteria, that is the smallest form of life, 5,000 genes. And if we look at the human genome, we discover that human genome has only 25,000 genes. And there is a kind of inconsistency, so the genetic determinism does not exist in the sense that we do not completely understand how it is possible that 25,000 genes may be able to or contain the information for synthesizing 100,000 proteins. So at least what we may say is that one gene should be able to synthesize 4 proteins. But the story is much more complex than this. Let's see, we started from Darwinian perspective, Darwinian evolutionary theory. I'd say the survival of the fittest. So the idea of Darwin was that natural selection selected was a mutation that were only was that guarantee the survival of those creatures of those living forms that were most fitted to the environment. Uh, However, you may recall we had a dramatic discussion between Darwin and Lamarck in the end of the 19th century. Lamarck introduced the concept of inheritance of the acquired traits or of the acquired characteristics, and this was something that Darwin was not uh, very able to explain with his theory, evolutionary theory, until we had until the discovery of DNA. But then some other aspects of evolution became apparent. In particular, it was clear that the surrounding condition in which life forms were developing may act in a significant way in the manifestation of the genetic information. So we had in the mid of 20th century the development of a new branch of science, that is epigenetic. Epigenetic, that is the study of changes in the phenotype. You may recall genotype is genetic information, so DNA, chromosome. Phenotype is how this genetic information and chromosomes are transformed into actual life characteristics. Epigenetic is any activity of gene regulation through chemical processes that do not involve changes in the DNA code, but may modify the phenotype of the individual and of the offspring. So this, if you want, solves a problem of 25,000 to 100,000 proteins, because there is the inclusion of external factors that may change in a significant way how these 25,000 proteins may be actually, the word is, may be actually transformed into chemical molecules, human manifestations. So there is a kind of gene expression control. During morphogenesis, we say totipotent stem cells, cellule staminali in Italian. Totipotent means stem cells that may be changing in every type of cells in the embryo, become the various pluripotent cell lines, for example, skins, liver, or bones, so cell lines for different types of organic tissues of the embryo, which in turn become fully differentiated cells. The question is where control comes from. The answer that comes from epigenetic also is control of gene expression comes from external environment, but is also linked to the so-called growth noise. So there is a kind of stochasticity due, for example, to cosmic radiation and so on. Point is, interestingly, that the gene expression control comes from external environment, is mediated through a kind of interface that links the external environment to what is happening to internal part of a cell, of a biological living cell. In particular, here we have a cutting, side cutting of a cell membrane, just to show you how complex this structure is. In cell membranes, you have a kind of liposomic, so fatty layers of biochemical molecules that subdivide the internal part of the cell from the external part of the cell. The connection between the internal part of the cell with the outside part of the cell is guaranteed by some kind of channels, the so-called that are channels made up of proteins, polysaccharides, and so on, that allow the passing of information in form of chemical molecules from the external part of the cell to the internal part of the cell. So this means, in our perspective, but one of the very relevant characteristics of living system is that of having a kind of interface in our language. In our class, we may call it a model, but mediates between the external environment and the internal environment. So in our perspective, we may say that a living object is an object able to perform a modeling activity, or able to implement a model of the external environment and to change and adapt its behavior according to the changement of the external environment. In this case, you may see that there is a striking and impressive analogy between a cell membrane, but it is a lipid, so a layer of different lipids structure crossed by kind of, if you want, exogenous or external or alien substances, the protein lipid channels that connect the outside to the inside through the phospholipid membranes. And this is the striking analogy with semiconductor. You may recall that semiconductor are wafers of silicon with some dragons here and there. Dragons are kind of metals like germanium and so on. And a semiconductor is the basis, technical basis on which we have constructed our computers. And it is quite impressive that most similar to the living object production of human intellect, which are computers, are based on some kind of semiconducting technology, like the cell membranes, so living objects are based on. So there is a striking analogy between semiconductors and living objects. So let's recast the question, where is intelligence? And I would say, in light of all the speech we have made up to here, that intelligence is mostly stored in the cognitive model for the interpretation of reality. So in a bacteria, intelligence is probably stored in the membrane. Cellular membrane that mediates the interaction between the external environment and the internal condition of a living cell. I recall you, external, perturbed and constant and random, internal, constant and controlled environment. Okay. Okay, let's conclude this lecture by observing that This story, we have left completely out the role of electromagnetic signaling. If you think that chemical signals, that is the propagation of chemical molecules containing some kind of information, move with the speed of centimeters per second, while electromagnetic signaling move with the speed of tenth of millions, 10,000 kilometers per second. If you are an organism of some billions of cells, what type of signaling will you use for communicate between trillions and trillions of cells, like large animals? Of course, chemical signal is the first choice, or at least how we understand it. Chemical signaling may be mediated through cell membranes that are quite complex biological structures with some similarity to semiconductors. But we cannot at all exclude the role of electromagnetic signaling. This is just to tell you, and just throw a stone in the stain, a stone in the pond of your intelligence, just to say that probably neglecting the effect of electromagnetic waves on the performance of biological system is not a good idea, because there is a type of signaling should have a very important role anyhow. Okay, so this is a time in which we are ready to switch from the biology to a quantum mechanic approach, that is the most modern part of physics. In order to switch from these two perspectives, we must make reference to a kind of materialistic prejudice, or the mechanicistic physician or Newtonian physicist uses hormones, cytokines, growth factor suppression, and what the role of electromagnetic fields. Up to now, this is still not well understood, or it is quite neglected. So when somebody tells you that electromagnetic fields do not have effects on the performance of living system, you should it should ring a kind of bell into your mind, saying, okay, this is not really acceptable to us. Okay, I think for now we may stop, and we shall meet on further. Do you have any questions?
No, Professor, thank you.
Okay. I wait for you. I wait here 10 minutes, and then I close the class, and we go on Thursday. On Thursday will be much more practical. So we'll go into the details of modeling physical.
Excuse me, can I ask a question?
Yes, please, I'm here.
About the topics that you have talked about until now.
Yes.
In the exam, how you gonna make? I just want to have an idea about how will be the exams in the questions. Your question regarding these topics in the exam.
Okay, my pleasure. Regarding these specific topics, will not be very heavy. The very important part of exams are the description of a model describing physical chemical and biological processes, but we shall start to discuss from next lecture. But these three lectures, that are introductory lectures, were useful just only to give you an idea of a perspective on how we modern ecologists look at living objects. So don't be worried too much, because the part that will be part of the exam are essentially classical process description. So models that will describe biological processes, as we will start seeing at starting from next week actually. The lecture of next Thursday will be an interaction between the spheres. So we'll start classifying biological, biochemical, physical processes.
Thank you so much.
You're welcome. Okay. See you on Thursday. Well.
Bye. Thank you.
Thank you. Goodbye.
Bye-bye. Thank you.
Bye. Thank you. Professor.